Welcome to EPG Pathshala. I am Dr. Sarita Nanda, Associate Professor in Biochemistry Department, Daulat Ram College, Delhi University, Delhi. In this module, we are going to learn about the neuronal cytoskeleton present in the human. The learning objectives of this module is that you will be introduced to the cytoskeleton, to the microtubules, to the characteristics of the microtubules in the neuronal cells, the microtubule associated proteins, the maps in the neuronal cells, the intermediate filaments, the microfilaments, and the regulation of their function of these cytoskeletal proteins. We are also going to learn about the molecular motors, the motor proteins which are associated with microfilaments and neurons, and motor proteins associated with microtubules, the role of molecular motors in these neurons, then interaction of the cytoskeletal elements, especially in the neuronal connections and in the glial cells, as well as alteration of cytoskeletal element in injury and neurodegenerative diseases. In this, we will first focus on the changes in the injury and regeneration, and thereafter on the neurodegenerative disease. So, what we see in this, that neurons are cells, as we all know, with unique structure and function. They are supported by cytoskeleton, which is made up of microfilaments, microtubules and intermediate filaments as all other non-neuronal cells. So, what is special about them? These cytoskeletal elements differ from the non-neuronal cells in the type of isoforms which are present here. Their organization is different and their participation in various functions are also different. So, if we look at the first element, the microtubules, what we see is the microtubules are like any other microtubules of the non-neuronal cells, but they are different from the non-neuronal cells, that they lie free in the cells and they are not emanating from the microtubule originating center as they do in other cells. And if we look at the polarity, then microtubule polarity is different in axons and dendrites. So, what we see that in axons they are arranged with the plus ends away from the cell body, all of them. On the other hand, half of the plus ends in the microtubules of the dendrites face away from the body and other half show plus ends towards the cell body. And moreover, the microtubules are composed of subunits alpha, beta, gamma as in all other cells, but their isoforms are exclusive to the brain. And moreover, they exhibit lot of modifications in form of acetylations, tyrosination, phosphorylations, and these modifications help in regulating their structure and function. Now, these neuronal microtubules exhibit greater stability as they do not depolymerize easily and maintain longer length than any other cell. Now, these microtubule associated proteins are also present with the neuronal cells, and these are maintaining their stability, interaction with other elements and providing them functions. The maps of the brain are as of other non-neuronal cells, we can see 1A, 1B, 2A, 2B, tau proteins, MAP3, 4, as well as the molecular motor proteins, kinesin, dynein and structural maps. And if we look at the function is the same, that they help in assembly, in disassembly, and they also help in transport of organelles, they also help in transport of neurotransmitters, and they also help to interact with other cytoskeletal elements. Now, the distribution of these maps is different in various parts of the neurons. The soma and the dendrites show the presence of MAP2, whereas the axons are rich with tau. Whereas MAP1A and 1B are present in the entire neuron soma, whereas dendrites and axons only have 1B, which is phosphorylated in axons only. It has been observed that there are 6 types of tau isoforms, and as in other non-neuronal cells, microtubules act as tracks on which membrane bound organelles and vesicles of neurotransmitters are transported in retrograde and anterograde manner. In anterograde movement, the movement is away from the cell body, whereas in the retrograde movement, the movement is in the reverse direction from tail to head. Now, if we look at the intermediate filaments, they are usually typical in each tissue, and they are therefore used as biomarkers. The intermediates in neurons are called neurofilaments, and those in the glial cells are called glial filaments. The NFs and the GFs, they possess several type of intermediate filaments which maintain the characteristic shape and functions. The NFs have projections from the sides, unlike the IFs of other cells, and they form loose network. On the other hand, the GFs do not show any projections like the cells of other parts of the body, and therefore, they are closely spaced network. The NFs are made up of 3 protein subunits: the high molecular weight, which is called as the NFH, which has a molecular weight of 180 to 200 kDa, the middle molecular weight protein, which is around 130 kDa to 170 kDa, and the low molecular weight NFL, which is 60 to 70 kDa protein. Now, all these 3 proteins, they intertwine to form a core protein at the amino terminal end. NFH and NFM possess a long terminal carboxy end which projects out as side arms. Now, these side arms can be phosphorylated, and this increases their density and repulsion. Vimentin is another type of intermediate filament which is present in the microglial cells in the neuronal and the glial precursor. Similarly, GFAP, that is the glial fibrillar acid proteins, are the IF which are present in the astrocytes and the Schwann cells. Some other IFs like alpha internexin, perforin and nestins have been seen in neurons and neuronal precursors. Alpha internexin is present in both CNS as well as PNS, and they are observed in developing brain rather than in the mature brain. Perforin is seen only in the peripheral nervous system. On the other hand, nestin is associated with development of the brain, and therefore, it can act as a marker of neurogenesis. Interestingly, some of the mature oligodendrocytes lack IF, which is under study and investigation. IFs are not only essential for cell survival, but they help to maintain the shape and the structure as they are one of the most stable cytoskeletal elements. The third element, that is the microfilaments, are also abundant in the nervous system, but their location is more concentrated near the plasma membrane and in the presynaptic terminals, in the spines of the dendrites and in the growth cones. They form networks and they are prominent cytoskeletal structures in the mature neurons. The microfilaments are of varying lengths. They are short in neurons, they are longer in growth cones, and they form bundles in filopodia and lamelipodia. And a special thing here is that they are not forming stress fibers like other non-neuronal cells of the body. Now, these MFs are also associated with several proteins which maintain their structure and their stability, and two of them, spectrin and encheirin, as seen in the RBC, are also seen in the neurons. And these are seen to be in the different isozymes, and these isozymes are present in dendrites and axons. Similarly, fimbrins also help to form actin bundles in the growth cone. Besides this, some other proteins like gelsolin, profilin, beta thymosin, etc., are also seen to be associated with neurofilaments. The microfilaments interact with the plasma protein and they restrict the movement of proteins from axons and dendrites. This maintains the characteristic of protein distribution in axons and dendrites. They also help the neurons and the glial cells to interact with the extracellular matrix to make a cell to cell interaction. They may also help to hold the cell organelles in the cell cortex. Interaction through tight junctions and focal adhesions are also helped by microfilaments. They are also helping in myelination, lamelipodia formation, filopodia formation, especially during the cell movement. The regenerating and the developing neurons have fast growing ends which are rich in microfilaments. Now, all these cytoskeletal elements are assisted by molecular motors. The molecular motors help to transport proteins, help to transport neurotransmitters, help to transport organelles in both anterograde movement, that is the movement from the soma to the axon, as well as in the retrograde manner, that is movement from axon towards the soma. Now, these molecular motors are various forms of myosins which work on actin tracts. Now, dynein and kinesin are the molecular motors which work on the microtubule tracts like in any other non-neuronal cells. Whereas, the motor proteins which are associated with microfilaments are several types of myosins which work with the microfilaments. These have been found to be myosin of various type, that is 1B, 2B, 5A, 6, 7A, 9A, 10, etc. The motor proteins associated with microtubules and neurons are the kinesin and dynein as in other cells. The kinesins associated with microtubules are kinesin 1, 2, 3, 4, 13, 14, and they help both in anterograde and retrograde movement. Whereas, the motor protein dynein is also associated with microtubule, and it is similar to dynein which is present in other tissue. And moreover, dynein is associated with dynactin, which is an adapter protein linking the dynein with microtubule. Dynactin have shown to be important in dendritic polarization and neurogenesis. So, this dynein is helping in the retrograde movement. So, now if when we look at the roles of molecular motors in neurons, then we see that they do various functions like movement of mitochondria within the cell, movement of cytoskeletal elements within the cell. They are also helping in polarization related factors to be transported. They also help the growth factors to interact with the receptors which are present at the plasma membrane, and their message moves in a retrograde manner towards the cell nucleus with the help of dynein. The neurotransmitters released at the synapse is also helped by the motors. The sensory organs like ears require myosin 1C, myosin 3 for the functioning of stereocili. A close interaction of cytoskeletal elements has been observed in a process of neurogenesis, interaction of neurons with the glial cells, as well as in the injury, regeneration and neuropathology. Now, if we look at the interaction in the neuronal connections, then when the synapse has to be formed, the growth cone present at the end of the terminal end grows towards the adjoining neuron. The neuroid projecting from the growth cone senses the direction in which it should grow. These growing ends has to arrange the cytoskeletal elements to achieve growth in the particular direction. While rearranging these elements, it may form pointed projections or web like projections like lamelypodia or filopodia. If we look at the interaction in the neuron glial cell connections, then what we see that the myelinated axons are rich in neurofilaments, and these are widely separated by spacer arms on the sides. The spaces are occupied by microtubules, organelles and short microfilaments. It is hypothesized that during myelination there is reorganization of these elements. The NFs are signaled to pack closely due to the reduction of phosphorylation of NFH and NFM tail domain. On the other hand, if we look at the changes in cytoskeletal element in neuropathology, then we see differences in injury and during regeneration as well as in the neurodegenerative disease. It has been observed that PNS neurons repair much faster and more effectively than the CNS neurons, and in some incidences CNS neurons may not repair at all. Following the injury in the CNS, the astrocytes proliferate to repair the area and result in forming a scar. This area is observed to be full of GFAP-IF bundles. This may help to repair, but the scar may also obstruct neuronal elongation and repair. The gene expression studies during regeneration of neurons have shown greater synthesis of microtubule and microfilament proteins rather than NFs, indicating greater participation of microtubule and MF in regeneration. If we look at the role of cytoskeleton in neurodegenerative disease, there have been speculations that some of the neurodegenerative diseases may arise due to malfunction of cytoskeletal elements which participate in vesicle or organelle movement in passing of the signals from the synapses. These changes may happen due to mutations in the genes. These mutated genes may cause defects in the functioning of mitochondria, which leads to oxidative stress and subsequently the DNA damage. Some of the cytoskeleton defects are seen to be associated with the following diseases like Alzheimer's disease, the Parkinson's disease and the amyotrophic lateral sclerosis ALS. In Alzheimer's disease, it is associated with accumulation of neurofibrillar tangles NFT. These aggregates are rich in MAPs called tau. It has been observed that tau in MAPs in NFT has excess phosphorylation. Therefore, this affects the polymerization of tubulin and therefore, the stability of the microtubule. In the Parkinson's disease, it is a disease which is associated with mutation of protein called synculin, which participates in formation of Lewy bodies and NFT like structures which are formed in the presynaptic terminals of the affected neuron. Another disease of the brain, that is amyotrophic lateral sclerosis ALS, the axonal transport of ALS affected neurons slows down. It is speculated to be associated with nitration of the tubulin molecules, which affects the microtubule polymerization and its interaction with the dynein motor proteins. ALS is also associated with mutation of the copper zinc superoxide dismutase. This leads to defect in cytoskeletal elements and vesicular transport protein. So, all these cytoskeletal changes may lead to loss in functions of motor neurons. So, students, let us summarize whatever we have learned, that first of all neurons have a unique structure and these are supported by the neuroskeletal elements, microtubule, microfilaments and interfilaments. Now, these elements give it structure, function and stability. The microtubules are similar to non-neuronal cells except for the few modifications. Some of the isoforms present in the neurons are specific for the brain and they may acquire some modifications by acetylations, phosphorylations, etc. These modifications may help to regulate their structure and function. Microtubules are stabilized by microtubule associated protein as in all other non-neuronal cells, but their isoforms and their arrangements differ in dendrites and axons. The microtubules and the axons are organized in a specific manner with the positive ends away from the soma, whereas the dendrites have the microtubules arranged in a mixed order, that is the positive end either away or towards the soma. Their stabilizing maps are also differentially distributed with map 1A, B and the various isoforms of tau found in the axons, whereas map 2A and 2B found in the dendrites. The intermediate filaments of neurons are of three kinds: NFL, NFM and NFH, whereas Vimentin and GFAP are present in Schwann cells and in the glial cells, and these can act as biomarkers of the brain tissue. The microfilaments are similar to non-neuronal cells which help in neurogenesis, plasticity, cell to cell junctions, restriction of movement of proteins in axons and dendrites. The motor proteins associated with cytoskeletal are similar to non-neuronal cells which participate in transport of protein organelles, neurotransmitters. There are several kinds of kinesins like N-kinesin, M-kinesin, C-kinesin and dynein which travel on the microtubule tracks. The dyneins support retrograde movement, whereas the kinesins support antrograde and retrograde movements. On the other hand, different kinds of myosins like 1B, 2, 5A, 6, 7A, 9A and 10 act as motor proteins which carry cargo of proteins, cytoskeletal elements on the microfilament tracts of the neurons. The cytoskeletal elements interact with each other in several events like synapse formation between two neurons, myelination of neurons by glial and the Schwann cells, and the research is going on to investigate the role of cytoskeletal elements in injury, regeneration and neurodegenerative diseases. Evidences show participation of glial cells and greater synthesis of microtubule and microfilament proteins during repair and regenerations. Several neurodegenerative diseases are associated with accumulation of aggregates rich in mutated proteins and also loss in motor function due to mutation of cytoskeletal proteins. Thanks for your attention.